Dr. Wittenberg started his studies at Harvard for undergrad. He went on to MD PhD program the other side of country, San Diego, University of California, where he got MD PhD degree. And then he went to WashU, where he did his neurology residency and then neuro rehab fellowship. I have to say that that is an unusual path for, he's a pioneer in neurology field to go to neuro rehab right from the residency. So, but that he was not done with neuro rehab training. He spent a couple years with Leo Cohen at NIH, path of the start of successful neurorehabilitation career. His first faculty position was also associated in Wake Forest, North Carolina, he established a fellowship, or he was a director of neurorehabilitation fellowship and was doing that for 6 years until he moved to University of Maryland, where he also was of staff of, he was a neurologist at the Maryland Baltimore VA. Dr. Wittenberg has associations with departments of medicine, gerontology, and neuroscience at University of Maryland. He was a director of, as I said, VA Maryland Exercise and Robotic Center of Excellence. So his research career started with his PhD, where he started, his experiments is actually on leeches. And as he mentions that there's a connection between neuroconnectivity work that he started in leeches and then moved to humans, where his focus is on understanding connectivity of motor control. His research activities currently in the last 20 years been on recovery of upper extremity motor function after stroke. He is interested in understanding motor control of reach and dexterity that is in normal aging and in Parkinsonism. Dr. Wittenberg is he is authored of 63 peer-reviewed papers. He has had continuous funding for about 20 years, mentored many scientists who went on to have their own distinguished scientific careers. So let me welcome Dr. Wittenberg. We're honored to have you here.
Great, thank you, Svetlana. It's very kind and maybe somewhat exaggerated introduction, but I think it does represent the sort of journey that I've been on, and I'm also honored very much by the invitation. I feel very much at home here. I've been here once before as part of a VA Center Directors meeting, and actually Bob Kirsch and everybody made me very much at home. I actually used an office for a day here, so it's like a little bit of a second home. And also, since moving to Pittsburgh, I think I'm part of this kind of Cleveland-Pittsburgh axis, which I know is very active, and we have I think many people who trained here in the lab. The RNEL is a big laboratory collective in neuroengineering in Pittsburgh, and so we're very fortunate to have that sort of two-way street. I really, this is actually my first talk at another institution since COVID times struck. So that's been over 2 years. So maybe a little bit out of practice, but I'm certainly very happy to be here. So just to introduce you to the overall themes of the talk, and actually it was useful that Svetlana mentioned the leeches. So I was working on sensory motor coordination in the leech, but in fact, it was really a talk I went to as an undergraduate by Ron Calabrese, who really showed how understanding a circuit could understand the motor pattern. At that point, he actually built a physical model with flashing lights that showed how the circuit connections reproduced the behavior that was observed. And that really has been what I've been working on ever since. In fact, I work currently on really three major themes, and I'm talking about one of them today, which is about circuits and behavior and plasticity. The other two are clinical trials in neurorehabilitation, looking at how to improve upper extremity motor function and predictors of response. And then the third is multimodal assistance for assessments and rehabilitation, including looking at sensory function and neglect. One other branch off what I'll talk about today has actually been trying to put together a database of TMS-derived knowledge about connectivity in the motor system. I probably won't have time to talk about that, but I may talk to some of you, whether here or not, about that in some individual meetings. I'm trying to get some feedback about that idea. So by now, I think you may have seen the slide. This represents a kind of chronological progression in that line of research of circuits and plasticity in recovery of function. So I do want to acknowledge many people who are involved, and this work covers a number of years and institutions. And it's interesting how when you move to different institutions, there are different strengths in terms of trainees, and at Maryland it was definitely the postdoctoral fellow program that was a Neidler sponsored program that really was very helpful. And also the collaboration with VA centers has been a theme at both places. I was able to do both a sabbatical and VA extended educational leave in Belgium, and I'll talk about that project in some extent. Their master's students were very helpful, and actually now at the Pittsburgh VA, I was able to bring Amy Boos, who's a occupational therapist and has a master's in engineering, to do interventions and coordination. And also collaborations, I didn't list them here, with the VA Center of Excellence there, HURL, with Michael Urban and Charlie Lafe and Gina McCarran has been very helpful, including even analyzing data acquired on sabbatical. And of course, the other students and volunteers are very much appreciated, and as well are the funding agencies. I do have one disclosure. I'm on the clinical advisory board of, it's MyoMo. They make the MyoPro, but I won't be talking about any work directly related to that. And then also an acknowledgement of the American Society of Neurorehabilitation. I was president and held other offices. I don't have any office in it now, but that really helped shape my career, and I think has a particular emphasis on career guidance. So I'd recommend it for anybody who doesn't know about it. They will have an in-person annual meeting, we hope, at the end of March, beginning of April in St. Louis, which is a great place for neurorehabilitation, which I haven't been to much since I left there, but it's definitely worth the visit. All right, so going into the first section, with the idea is that my goal, as I think many others here, is better rehabilitation after stroke. So how do we make rehabilitation better? If we don't know about the circuits in brain and spinal cord, how do we go about repairing them? It could be a lot of trial and error, but there's such a huge parameter space. It's really not really practical to just try different things and then see what works. So we'd really like to know how the circuits work. And TMS, transcranial magnetic stimulation, I'm not going to give a background on the technique. I think by now most people have heard of it or used it. It's a method that's very useful in establishing the dynamics of connections, because the pulse of stimulation is well less than a millisecond, even though it has effects that last for several milliseconds. But by being able to control a stimulus, it gives you a different tool to understand the causal relationships of activity in the nervous system. And so the first study will actually focus on plasticity in the primary motor cortex at M1, and then we'll work towards looking at other areas. But circuitry involves both intracortical circuitry within a region and also interregional. There are connections. So it's still about circuits in a way. So this project on looking at TMS, evoked robotic movements, and plasticity, this was an idea that that really came up when I first became aware of rehabilitation robotics, and I realized that they were a very good framework for studying the plasticity of movements related to practice. So you can regiment practice with a robot, you can measure movements very, very precisely and measure every movement with a robot. There's a paradigm that was developed in Leo Cohn's lab called the thumb paradigm, in which TMS was used to evoke thumb movements, and TMS over the motor cortex will evoke movements in everybody of the thumb that will have one dominant direction. And if the person practices in the opposite direction, then the TMS movements can be reversed. So it's a model system for studying the effect of practice on brain circuits. And I listed here some of the questions we have is since people do improve after practice, what brain areas are responsible for that improvement? And in stroke, why is the improvement inadequate in so many people? Some people it works well, and some it doesn't. And then in the people who it doesn't work, can you augment the effects with other methods like brain stimulation? Can combinations of brain stimulation and robotic practice result in useful plasticity where it wouldn't with one alone? And then, of course, are there any undesirable effects of sort of hybrid stimulation treatments? Maybe the TMS would interfere with movement. So here you see a basic experimental design for a plasticity experiment on the lower row. You have people with a baseline, and you can do baseline motor assessment. You can also do TMS, basic TMS measures of TMS evoked movements. And here you see one of the earlier versions of the MIT planar robot, shoulder elbow robot, with the participants holding the handle of this end effector robot. And so after you establish what movements are evoked by TMS, then the participant practices in the opposite direction. And this practice can be fractionated, so you can have probe trials where you measure the TMS evoked responses or other things in between practice, and then you can measure retention at the end. So because we want people to practice in one direction only, they practice against a spring field. So it's as if there were a spring attached to the center of the robot handle. There isn't one. There's a force that's calculated by the robot and provides a return to the center. So people are making forceful movements against the spring field in one direction and then returning to the center. So the outcome parameters measured in this kind of work are based on the position of the robot handle. If you measure the velocity of the handle, then there's a point of maximum velocity of these TMS evoked movements. So these are TMS evoked movements. They have a rapid acceleration and then a deceleration, and then the spring field returning the handle to the center. So the TMS evoked part of the movement is this ballistic movement. Here they are in 2 dimensions, and amplitude and direction of the movement to the point of maximum velocity represents a good approximation of the torque that's developed by the stimulation. And then there's some baseline direction and amplitude of movement. The people are practicing, as I said, in the opposite direction, and then at the probe trials after practice, the TMS evoked vector is different. You can measure the displacement of that point of peak velocity. I won't show that data. I'll show directional and amplitude data of the vectors. So I'm skipping ahead a little bit. I'm not telling you the whole story. We'd already established what TMS evoked movements are like, what practice-related plasticity is like, and in fact, there is directional plasticity after practice. It's not 180 degrees. It tends to be closer to about 60 degrees, and then we wanted to see if we could have an interaction of the practice with TMS, and so the study was designed to stimulate once every 10 seconds. It was actually about it was exactly half of the trials. People would practice every 5 seconds. Stimulation was every 10 seconds, really just to make sure that we always had the stimulator was always recharged at the time of stimulation, but we had good practice effects. And we were particularly interested in the timing. So previous combinations of stimulation with practice had involved EMG triggered movement, where the stimulation is triggered by the activation of the muscles, which is good for synchronizing the stimulation with movement, but bad if you think that there's a LTP-like effect in which the presynaptic activity really needs to precede postsynaptic activity in the target neurons that are producing movement. So we actually tested 3 timing paradigms. One, which was a late reaction time period. So we measured reaction time for target presentation of a target to movement in the training direction, and then triggered the simulation 150 milliseconds after, which is earlier than the shortest reaction time. There was also the EMG triggered as a comparison, and then we did have a sham control, but a better control was actually random stimulation, where it was purely measuring the effect of timing, and so the stimulation could occur anytime during the preparation or movement period. So these results show the effects on the directional plasticity and the amplitude plasticity for the different probe trials. So you see the 4 different trials after practice started, and these represent change. So I know it's a little bit small, but what you see is that in all 3 conditions, there's a change in direction that's significantly different from the baseline condition. There's no differences among the groups. So and this was actually a fairly similar amount of directional plasticity to what we saw before. In experiments without stimulation. Interestingly, though, this was a group that was an older group that was with an average age that was close to 60, meant to be a age-matched group for potential stroke studies, of which we did some. So we were able to get this kind of plasticity even in an older group. But then the interesting thing here is the amplitude change. So here with the pre-movement simulation, you see an almost 80% increase in the amplitude of TMS evoked movements, and much less with the EMG triggered and random sort of in the middle. So there was a strong effect of the stimulation timing on the amplitude of movement. This is just so it's nice to see what the actual data looks like. These are real individual motor evoked potentials from four muscles. It's biceps, triceps, anterior deltoid, posterior deltoid. So for prime movers for planar arm movements, and along with actually the directions of TMS, individual TMS evoked movements. But I think you can see a striking increase in MEP for this pre-movement stimulation example, a striking decrease for the EMG triggered, and no change. Obviously, we picked best examples from the group, but these are real examples. For the MEP plasticity, so before the graph I showed you was the movement's amplitude and direction. If you actually look at the motor evoked potentials, here it gets a little more complicated because the directions that people practiced were different. They were depending on their individual TMS evoked movements, which do represent a range really around the circle. So we categorized the muscles into elbow agonists and antagonists, shoulder agonists and antagonists for the practiced movement. But it doesn't really matter when you're looking at this graph because it's actually the same pattern that for the pre-movement stimulation condition, there was an increase in motor evoked potentials in all muscles, not just the agonists. For the most part, in the EMG triggered stimulation, there was a decrease in both agonists and antagonists. So in a sort of ideal world, if you're trying to explain the directional plasticity, would be by a change in the balance of these motor evoked potentials. There are some limitations in just how much of the muscle activity you can measure from surface EMG, how many muscles. In a sense, the motor evoked potentials had to be somewhat specific to the action of movements. We just didn't see it in this, and so it actually implies that there's a more general effect on muscle activation with these two timings, which in itself is a very powerful difference. It's only about 100 millisecond difference in the actual timing of these two stimulations, but it matters for these responses. And so the utility here is that there are maybe points in which you do want to suppress certain types of activity and enhance others, and here it's a timing that's more specific than the direction of movements being practiced. So I'll sum up this part of the talk with saying that we found that stimulation affects the practice effects on TMS evoked movement in a timing-dependent manner. In a sense, the balance of synergies, or however you want to think about the sort of pattern of muscle activation, we couldn't show how that was affected by stimulation time, but it does provide this means to enhance practice effects in stroke. And in fact, we did do pilot work in which we combined stimulation with practice in stroke patients and actually were able to change the kinematics in a useful way. There's another limitation in doing that, which kind of affected some of the future studies. We can talk about that if people have questions. This work has been published. So then moving on to Belgium. So in doing this work, I really recognized that I had a certain hole in my own education in terms of how to do motor control experiments and analyze them. And so there was a laboratory at the Catholic University of Leuven, which is actually the largest university in the Low Countries. This was the residence area there where we lived, that actually had strengths in both motor control, aging, and functional imaging. And so that's where I went for a year. And so just to introduce the ideas about this project, but I think also more generally in thinking about movement planning and execution brain areas. So it's the same on the left and right. On the right is my attempt to summarize in just a few words what these brain areas do. They're almost certainly not completely correct, or maybe even wrong. And I think maybe some people in the audience know there's still a lot of debate and discussion about what the basal ganglia actually do. Here shown just by the output of the globus pallidus pars interna, maybe have something to do with action selection or vigor. The thalamus is an important relay, and with the cerebellum provides input to areas mostly through the thalamus. But you can't avoid that in primates. The primary motor cortex M1 is the most important area for at least distal fractionated movements of the limbs, and provides the bulk of the corticospinal tract. So when there's damage to that area, these other areas may influence output, but often they're influencing output of that damaged primary motor cortex. And right, so some of these areas we'll see that we can actually characterize their role in reaching behavior, and also their timing of their role, because this diagram doesn't emphasize the dynamics that we know are important to movement. So, going into Belgium, I already planned one study, had hypotheses for the study, although the hypotheses are based on that sort of knowledge about what we think brain areas do. So the idea was to test the role in reaching behavior without anti-gravity support, which actually many therapists have looked at these robots that provide anti-gravity support and said, well, this doesn't represent the real world. Let's see what the control of movement is when you don't have that type of support. There may be different circuitry. And so we're going to test the role of these different areas with short trains of TMS. So short trains of TMS can be used to interfere with a region's activity. It's complicated because it is stimulation, but it also introduces a more longer-lasting inhibition and also noise into an area. So it has generally an interfering effect on movement for several hundred milliseconds, or between 100 to a few hundred milliseconds. So the hypotheses were that supplementary motor area may have an earlier, more postural anti-gravity role than a ballistic one. That when I say PMA in this context, it's dorsal premotor area, may have more of a role in aiming and visual motor coordination. The M1 was included as a positive control. If we didn't have some effect on movement with the stimulation of M1, there'd be a problem with the method. And then it's always a problem figuring out a good control region in TMS studies. But in this case, we used the ipsilateral dorsolateral prefrontal area. So the right hand was being moved, and so this is the right dorsolateral prefrontal cortex, not because it's not connected to other areas, but it's more remotely connected. And then we thought that there would be a effect on motor output that would depend on timing, with the idea that there's more response inhibition early on, and then more of an ongoing motor command during the execution phase. So by measuring trajectories in three-dimensional space, we could measure things like loss of posture and variability, and of course direction and amplitude of movement. So this was the setup. It was sort of old school in a way, a physical board with LEDs, actually controlled by the parallel port of an older PC. But there were these three targets. I'll talk more about that. We had lots of wireless EMG, reflective markers. The whole room was a motion capture room. And so you already saw the three reach targets. They're vertically arranged so that there'd be different effects of gravity, but sort of the simplest possible arrangement for finding those effects. The LEDs would show the target in a pseudo-random fashion. There's never a surprise about what the target would be. There was a ready queue that always included target information, so we weren't trying to measure change in plans, which is also interesting, but would make things more complicated. The TMS pulses were just two in this case, 100 milliseconds apart, and they started either 50 milliseconds before, or 100 or 250 milliseconds after the go cue to move to the cue target. And we adjusted the simulation strength so that we tried to reduce, didn't actually completely eliminate motor evoked potentials. And that's the methods. So here you see the setup again. People started at a sort of neutral position in front of them and were reaching about 15 centimeters ahead to this target board. So these are real reaches in a younger and older individual, showing the actual facts on average that there was more variability in the older individuals reaching. So there was this preparation cue that lasted a second, a somewhat randomized delay, so they had to wait for the go cue, and then they had 1 second to reach, so there wasn't a rush to reach. They had to just reach the board and learn by practice to get there in about a second, then hold it there for a second. So why do we do younger and older individuals? And as in many studies, if you're interested in eventually studying stroke, you'd want a population that's better matched to the stroke population, but it's often easier to pilot and develop the method in younger research volunteers. And then it's also interesting to look at the difference between the two. So I'll show you the remaining. The following slides will have the younger on the left and the older group on the right. This is actually showing reaction times for stimulation in the 4 different areas, with the no stimulation trials first, and then the 50 millisecond before, and then the 100 millisecond and 250 after the go cue. And this is important to look at. It's really an important technical issue with TMS experiments when you combine a behavior and TMS, is that there's an effect on reaction time of TMS, so that the 2 earlier stimulation times decreased the reaction time, and right. So and the 250 milliseconds didn't. So that becomes important as we look later. The analysis of the effect of regional effects of stimulation was done with the looking at the timing first, and then all of the data. And surprisingly, there wasn't a strong effect of timing. There wasn't a significant effect of timing on most parameters. So, but because of a concern that we could be altering the actual quality of movement just by having an earlier reaction time, a different point in the motor preparation phase, the following slides will just show the 250 millisecond stimulation data in which we didn't change reaction time. So this shows the movement away from the home location, just 100 milliseconds after movement onset. As everything that you'll see here is normalized to the largest effect of stimulation on magnitude in the whole group, in the individual, so within an individual, but not for the particular stimulation location. So you have M1, premotor, SMA, and our control region. And so if you look at the highest value here, you see that the premotor stimulation for the lower target had the largest effect, with a highest increase in amplitude even this early after movement onset. So this is the lower target, middle target, and upper target. So the largest effect was actually for the smallest movement and the least anti-gravity movement. In the older group, there was a similar pattern, but less target effect. So just so you see it with these graphically for an example, what the magnitude of the reach vector is, that the motor and premotor areas had similar but not the same effects on increasing reach magnitude, and much larger than the effects of SMA and the prefrontal cortex. If you actually just focus in on that early area, no, I don't have the focus there. Okay. It also affects the reach angle. Now here, the upper target is the one that's most affected with a deviation from a straight line. Again, with the largest effect in the young group on premotor area. Less differentiation of effect, but a similar pattern in the older group. More deviation from a straight line path for the upper target, again premotor in the younger group, and then focusing on that deviation, you can now see that there's, again, this is a single example, but a difference between premotor and motor cortex in the actual angle of movement. So these premotor effects, we're convinced, are specific to the region and not due to some overflow of activity induced in the motor cortex. Let's see. Right. And then this is actually the same data as in the previous slide, but just an early inset, so you can see the differentiation of activity better. The point of maximum velocity is actually here. So you're seeing a big deviation even before the full movement gets developed. And the reach path curvature is affected as well. So it's a similar pattern. What's interesting is that even though you're seeing this larger effect in the young group, actually measures of endpoints, which I'm not showing you, variability and accuracy of endpoints were actually better in the younger group than the older group. So there was a greater effect of the stimulation and more regionally specific effect of the stimulation in the younger group, but a better adaptation to that, an ability to correct that by the time they reach the target, also in the younger group. And so here are some real examples of the deviations of the reach path, and you see that these are significant deviations in path, and again differentiating between M1 here in green and premotor in red. And the averages are the thick bars, so you can see much more of a sort of upward movement in the M1 and premotor group stimulation. So I know it's a lot of individual data there. I'll try to sum that up for you. Not everything, a lot of the differences came out through the statistical analysis and not by inspection, as it should be. So we found actually kinematic effects of stimulation early and late in the reach cycle for stimulation that was meant to interfere with about 200 milliseconds of activity in the brain region being stimulated. There were actually strong interactions of the target height with age and with the region of stimulation. The effect of stimulating both the M1 and premotor areas were actually to increase motor output and deviation. So this wasn't the planned motor activity. It was a different motor activity, but more of it. So it didn't actually, it could have turned out that people had a pause in movement, and that in fact was not the effect. So the way I interpret this is actually a disinhibitory effect that's at least a large number of the interactions of M1 and PMA are known to be inhibitory, and so we think we decrease the shaping effect of M1 on the movement by the stimulation, and the stimulation worked at any timing that we tried, so it was an ongoing effect. SMA stimulation was somewhat disappointing. We didn't see an effect on anti-gravity posture. It may not be there. That could be the reason. It can also be because SMA is somewhat deeper in the brain, that we didn't have an optimal way of stimulating that area. So I wouldn't make any strong conclusions from that. But again, just the age effects, we saw stronger effects of stimulation in many parameters in the younger group, but more effects on accuracy in the older group. This is the TMS work is published in the paper that Mike Urban was the first author on. Actually just got word yesterday that this publication on the age effects on trajectory was actually accepted for publication yesterday. So it'll be coming out soon, but you can read it on bioRxiv. All right, so the last section is based partly on those two preceding projects, on looking at non-primary motor areas, their effect on reaching, and their potential for improving plasticity after stroke. So the goal of this project, which at this stage we've completed a pilot and now have funding from the VA to study in stroke patients with a variety of methods, which I'll tell you about. So the idea is to quantify the effects of disrupting activity in both the dorsal and ventral premotor areas in both hemispheres. So not making assumptions about which one is doing which in the hemispheres, especially because we're studying people with internal capsule stroke. So we made what I think is a somewhat bold decision to not try to include large numbers of stroke patients, but really a specific stroke type as a model for subcortical damage that affects the output of the primary motor cortex. Then we also quantified the connectivity of these regions with M1 in both normal participants and in people who've had this type of stroke using MRI, and then the last part of it is to evaluate the effects of time stimulation of these premotor regions with practice, and with an idea that we can actually personalize the stimulation method and practice method to improve those improve movement, voluntary volitional arm movement, but using what we know about the connectivity of those areas. So, as I said, there's two types of participants, control and subcortical stroke, that have hemiparesis. We're now using the Kinarm robot, which is another planar robot. It's an exoskeleton robot. It's more designed to be useful in research. I think some of you are probably familiar with it. We have just two targets, ahead and behind of the initial location. Now we're actually doing reaches to remembered targets, so we want to make it difficult and not have the complications of potential ongoing visual feedback that, of course, people still have proprioceptive feedback. And we're simulating at a constant intensity that's below motor threshold. So we don't have the complication of motor evoked potentials or differences or heterogeneity in stimulation. So we have now 5 locations, the bilateral premotor areas of these two types, and then a midline, parietal midline area as our new control region. But then also testing different timings. And then, as I said, we're doing MRI with resting states, connectivity in the pilots, but we'll be doing more in the actual upcoming study. So this shows the Kinarm setup. This is how you can take away visual feedback. There's a screen here that can be opened and closed, so people can or not see their hands. They see a visual display, just like what you see here. You can take away the feedback of where the hand is, but you can show targets. So now we're looking at these circuits, the two premotor regions and primary motor cortex on each side, with people having a lesion that affects the output of these 3 regions potentially on one side. And of course, there are multiple connections, so the circuitry is still complicated. The effects of the interhemispheric effects are often inhibitory. That's shown by these little ball connections here, but complicated because disinhibition is also a form of excitation in a way of getting activity in another area. So for the pilot study, I'm not going to go into such detail, but we had these two normal participants. We're just calling the left side affected because that's the side that was moving the right hand. Obviously, it's not affected. And then we had two stroke patients with very small subcortical strokes, but with both a left and a right hemiparesis. So we don't have the data fully analyzed, and some of the methods are still being developed, but just in a basic measure of success rate. So this is actually a challenging task to actually reach to a remembered target. The four participants are represented by the four bars here. So the first two controls and the two stroke patients. The control region for stimulation is here, and you can see at least the affected ventral premotor area actually markedly reduces the success rate of getting to the target, and the other regions are sort of in between. So I think it's helpful just looking at the trajectories. Here's a control participant moving to a forward target and a backward target from a starting location. These paths are very, very straight in this case. If you look at the control stimulation region, this fourth stroke affected individual had very looping trajectories, but were actually made worse by stimulation of their right ventral premotor area, and may have been able also changed in their straightness in a positive way by left dorsal premotor area. So a complex interaction. The MRI data, so if people are familiar with resting state connectivity, I think this has become a common method in which you look at the correlation of bolt activity in the MRI for a seed region that represents the region of interest. So we developed regions of interest in dorsal and ventral premotor areas and primary motor cortex, and then thresholded these maps to a fairly stringent criterion to look at what the network is that's connected to each area. And the great thing about this method for a stroke patient is that they don't have to do a task in the scanner, so they can concentrate on keeping their head still, and head motion affects activity. In fact, this little fringe of activity here is likely head motion related. But what you're seeing here is the networks connected to the seed regions of M1, dorsal premotor, and ventral premotor, with the left hemisphere's network. Again, left is on the right side, shown in blue, and the right network shown in red. So if you look first at the control region, you can see that left and right have each have their own representation and areas of connectivity. Dorsal premotor, again, has this kind of mirror image of the two right and left regions, as does ventral premotor. This person had a stroke in this region, this subcortical region, and you can see complete disruption of the right ventral premotor network, despite the fact that the lesion does not include that area, and a more robust network in the dorsal premotor area. Also, loss of the affected side motor activity. So really a sort of complex network change that's not all loss of connectivity. And again, this is just examples. So the fact is, in the upcoming project, we're actually looking also at task-related connectivity, so and anatomical connectivity. So we'll have a lot of information about each participant, and we'll be able to see how each of these measures compare to the other, including the TMS effects on behavior. So that's basically what I said here. Interpreting those results of the comparing the two types of connectivity kind of created this matrix of potential results. So you can find that a particular non-primary brain region or premotor region has an increased or decreased effect after stroke compared to controls. You can also find MRI-based connectivity, as I just sort of illustrated, both increase or decrease compared to controls. So this diagonal is fairly easy to explain. If you see an increased TMS effect and increased connectivity, this means there's a functional reorganization that supports behavioral recovery. If, on the other hand, there's less TMS effect and less connectivity based on TMI, that represents a regional disconnection based on the lesion. But the other rows are also, the other cells are also possible. You can actually have decreased TMS, MRI-based resting state connectivity, and have an increased TMS effect. So, as I sort of illustrated here, that could represent a kind of Learn's non-use, that the correlations just with kind of idling behavior of the brain are actually lower. Those connections have become disused, but they still have a potential with stimulation. On the other hand, you could have increased connectivity but decreased TMS effect, which means that there's a sort of attempted compensation. The connection is increased, but the damage to the output system is too great for that connection to have an important functional role. And that this is just really starting to think about these things. But I think they represent a pointer towards how to try to correct the system. In general, you're trying to achieve more normality, but that normality may require help in terms of both practice and stimulation. All right, so this is just a chance for me to try to sum this up, and I think you probably got an impression that it all isn't of a sort of unitary value in terms of the take-home message, but some of the points are that the primary motor cortex, M1, is still very central to voluntary motor activity, but it's part of a circuit that can be influenced by stimulation and potentially stimulation in connection with practice. And that's the response of certain functional reorganization can be non-optimal in sort of the natural state, and this sort of non-optimality can be potentially addressed with rehabilitation methods. I think also that there's a sort of larger significance to some of the findings in terms of work on brain-computer interface and FES approaches, and in knowing where in the brain you might want to decode activity, or on the other hand, potentially stimulate to get desired activity. So thank you for your attention. I hope hopefully we'll have some time for questions, and thanks. All right, so now how are we doing? Questions from the chat or from here?
If you do have a question here, please make sure that we use the microphone so that people online can hear us. And I think that should work. Yes. And I'm looking in the chat right now.
I have one question. I was just curious. I'm not an expert at TMS by any means, but during an experiment or during a whatever you call them, the session, does the effect of TMS have some adaptive change on the responses? I mean, are they absolutely uniform from the first event till the last, or does it change over time? Is it or is there some adaptive change?
Right. So for those in the room who didn't hear the question, but the question is whether there's a kind of temporal effect of TMS on responses. So yes and no. I mean, there's a if you stimulate at a low enough rate, and generally once every 5 seconds to once every 10 seconds, responses are generally very stable. So without pairing with something else, but it's really the effects of practice and continued stimulation with practice, which do have changes over time. In the data I showed you actually was not that. There weren't that many temporal trends during the practice sessions. Without simulation, we actually saw more of a sort of temporal trends. So I think the overall impression is that stimulation is actually increasing the rapidity of the plasticity, but but I think for low rate TMS, there's not a sort of temporal trend. For our TMS, for repetitive stimulation or high rate, you can induce changes, both positive and negative, in the region. That's another sort of tool in the toolbox, as it were.
I have a question. So I work with sensation and spinal cord injuries, and I do know that there is maladaptive neuroplasticity in the somatosensory cortex for people with spinal cord and stroke patients, especially when afferent signals aren't sent to S1. My question is, in the motor areas of the brain, do you also see maladaptive neuroplasticity? And if so, how does that affect the reaction time of the system you are trying to look at?
The question is one about maladaptive plasticity in the motor system. So that can be a hard question to answer because, to test whether the plasticity is maladaptive, you would have to see, like, if you remove that, would things be better adapted? So from a causal state, it can be hard. But there are things that seem to be maladaptive. There also see, another idea in motor learning is one of scaffolding, the fact that sometimes things change as you get towards a response. So one thing you see after stroke is disinhibition, reduced paired pulse inhibition, which then goes more towards normal later, although you can see people with persistent hyperinhibition in an area. So that would represent the sort of maladaptive plasticity. There are all sorts of debates about spasticity and whether that's a sort of maladaptive spinal plasticity. I guess I don't, in my own thinking, I don't really think too much about maladaptive plasticity. I think about sort of inadequate plasticity and how to supplement it.
Nice presentation. I have two questions. The first one is about the study that you looked at I think you looked at the elbow extensors and flexors, and you look at the TMS timing.
Right.
I wasn't able to understand exactly how much the timing affected the activity of these muscles. Was that exactly the same level of was it like improve both of them or decrease both of them? I wasn't able to see that.
Okay, yeah, sorry, it was a little small. So the question is about the effect on the extent of the agonist and agonist of the two joints for in the experiment with the timing. So I mean, there were individual little differences, but the big picture was that the late reaction time stimulation increased the motor evoked potential in all four muscles, elbow and shoulder agonists and antagonists, and decreased it in when it was the EMG triggered stimulation. So, if you look at the original paradigm, the thumb paradigm, the plasticity isn't completely explained by the difference in MEP and agonists and antagonists. And as I was saying, you really have, in some ways, the muscle activity has to equal the movement in some way, right? I mean, it's being not exactly, but the forces are being transmitted into the part of the body, and there isn't some separate force. But in this case, even though we got a change in the movement's direction, the increase or decrease of muscle activity in those muscles was fairly uniform.
And my second question is about your Belgium study. Do you think if you have, I don't know the cohort that you work with the older participants, but I was wondering if you have people who are active and actually exercising, would you think that the difference between the young and the older will be less?
That's a great question. So that's about the young and old differences. And so, I was sensitive to that. So we did a survey in the study about people's activities because, this came up in the earlier study in the robot is, what if people are really practicing something? What if they're, fencers or musicians or something? So we really looked and it was really interesting. In our older group in Belgium, everybody was doing some kind of physical activity for the most part. I mean, they were all cyclists and hikers and dancers and things like that. So it was generally a pretty healthy older group. with relatively few exceptions. So I mean, it's good in a way, but we're looking at a sort of age effect that wasn't, it didn't represent people with metabolic syndrome, with heart disease, who were sedentary. These are all fairly active people. It would be interesting to do that in a different aging group. I think that's one of the big unanswered questions about aging in the brain. Is why is there this change in inhibition? I mean, that comes up in study after study, that there's reduced inhibition, larger areas of activation related to a task, all sorts of things just related to a sort of, just a change in circuitry. It could be compensatory for musculoskeletal issues. And one thing I didn't tell you about, we found in the older group, there's more co-contraction of biceps and triceps in that group compared to the younger group. But it was, as close as we could tell, a fairly healthy, active group.
Many of the interventional studies in stroke rehab seem to suggest that there's an effect of time post-stroke on outcomes and severity of motor impairment also on outcomes. Are you able to look at that as a factor in some of the experiments you were doing to see if you also can identify an effect of those two factors?
Yeah, so the effect of time after stroke is very interesting, and work I didn't show you back further back, we actually looked at TMS and MRI changes with time as early as a few days after a stroke, and it's totally different. Things are really changing there. All of these studies were in when they were stroke patients. They were chronic enough that there wasn't a difference. And I think there's this debate: is there a difference between someone who is 6 months after stroke versus 5 years after stroke? I don't really think there are really appreciable differences. I mean, we've seen some of our best responders in stroke rehabilitation trials have been 10 years after stroke. They also may be younger at the time of the stroke, but there are very important temporal things that happen. And I think you may know that the Dromerick et al. Study came out in PNAS recently, showing a particular sort of sweet spot for intensive therapy in a few months after stroke. And I think that's very important for future interventions and plans for the mechanisms. I think there's a benefit to going with a chronic state in which things aren't changing over time.
Thank you for your talk, Dr. Wittenberg. I'm also not well versed in TMS, but we do see effects in those specific tasks. So my question is more of, are those changes in plasticity, can they be translated to other tasks to see that the whole practice doesn't affect, any of the long-term potentiation? Right.
So that's a great question about people have heard of the translation of like TMS-related plasticity into tasks. So I mean, that's been an issue of debate, and it's been a criticism of the sort of stroke paradigm as the thumb paradigm as a model, for looking at interesting plasticity. So, we've done this as part of the work with robotic reaching, and in the normal participants, you really don't have an effect on their voluntary movement. And that's in some ways not too surprising. I mean, already people are good at doing reaching movements, and you could probably only make it worse, by inducing some plasticity, and people compensate for that if we have done that. But we have had limited experience with stroke patients where we can actually improve kinematics with the pairing of practice and stimulation. So I think it represents an important sort of translational pipeline, is that you can show a physiological change that, and then the next step is you have to show that it actually affects a laboratory measure of motor function. Then does it affect, home motor function? You can't try to hit a home run every time. To use a baseball analogy, you can't, do TMS pairing with reaching movements and then expect that someone's stroke impact scale, or that they report their quality of life is, improved 100%. We have to sort of like work on what we can measure accurately first, and then, if we develop a method that works, then look at some of those other features.
Thank you, Dr. Wittenberg. Appreciate you coming here. A terrific talk.
Thanks.
As a small token of our appreciation, it's probably Cleveland-related things, right?
Great.
So thank you, and thank you everyone for attending.